The aim of this procedure is to investigate the functional relationships between bacterial genes and pathways. This is accomplished by first preparing arrays of Escherichia coli single mutant strains that will be conjugated or mated. The second step is to conjugate the single mutants in an arrayed format on plates. Next, the double mutants are selected. The final step is to image the double mutant plates and quantize double mutant colonies to determine strain fitness. Ultimately, the colony size data are analyzed to calculate genetic interaction scores and identify functionally interacting genes, pathways, and processes.
The main advantage of this technique over existing methods like random mutagenesis is that many individual double mutant strains can be created. Moreover, the interactions between many genes can be assessed simultaneously, irrespective of gene size, essentiality, or single gene mutant fitness.
This method can help answer key questions in the systems biology field, such as those relating to gene functions and functional crosstalk between pathways and processes.
The implications of this technique extend toward therapy of bacterial infections because knowledge about most functionally central genes and processes, as well as the understanding of which combinations of bacterial genes, pathways, and processes, when perturbed, improve or reduce bacterial fitness, will help develop the most effective intervention strategies.
To begin this protocol, two-step nested PCR amplification is employed to replace the target open reading frame with a chloramphenicol resistance marker from the PKD3 plasmid as described in the written procedure. Confirm